Can you hear me all there, or can you hear me?
Yes, yes, your voice audible, Dr. Pradhan.
Can you see the slide now?
Yes, Dr. Pradhan, can you please put it to full screen?
Can you see now?
Yes, yes, yes.
Okay. Thank you, Dr. Susma, for the invitation and also the very nice introduction. Um, I live here in Uppsala almost 10 years now. Each time flies, you know that this is a beautiful city of Sweden. It is just near to Stockholm, and it is Uppsala University, which is the one of the oldest university. It was founded in 1477, and sometimes it is also called as a student city because so many students are there in the city. So this is so beautiful nowadays, the weather, and you can see my screen. It is even more beautiful in this time of the year. So let's get back to the topic. This ultra-sensitive and specific protein detection through merger of these two powerful technology. One is NanoPro and
On
another. Sorry, is it audible?
Yes, yes, yes.
Uh, so these two powerful technology for the protein detection. One is NanoPro and another one is proximity ligation assay. So today's I will talk a little bit about this Western blot, then go to NanoPro technology, which is also called as Capillary Western. And then we'll discuss about the proximity ligation assay, or in short form that is called the PLA, which is a popular name. And then how we merge this both technology, NanoPro combined with the PLA, and what are the advantage and the disadvantage of each of the all the techniques. So let's go to the conventional Western blot. And what are the challenges? So if you see, this is a conventional like Western blot. It is transferred SDS page, and then all the proteins are transferred into the membrane. And this membrane is stained with the protein ONCUS stain before you proceed to the development or the further step, incubation of the primary antibodies, secondary antibodies, and the development. So if you see all the lanes here, very nicely transferred, all the proteins. All this, like I think 20 lanes, it was very nicely done. Everything is transferred equally uh, charged in all the lanes. But the problem is, if you see these two spots, one is here and another one is here. Except these two spots, there is the problem here. That part is not transferred. The protein is not transferred in these two as a bubble. And even if you do, and even if you are an experienced person having done so many times Western blot or the transfer of the proteins from the SDS page to the membrane, but still it is, you can get, if you are lucky, then you don't get the bubbles. But if you are not lucky, then it is a kind of, this is a heat and trial method. So every time you do so cautiously, and then still you get these bubbles. Okay, then this is one of the challenges in conventional Western blot. And then this is the second part of the picture where we have done this, take two primary cell lines treated with the growth factors in different times, different time points, 0 minute, 7 minute, up to 24 hours, and collected all the cells, lyse it, and then run for the same Western blot. And then probed with the this MEP kinase, one to phosphorylated protein, and then this is the total MEP kinase protein, and also the housekeeping protein. And this looks like really fantastic result. As soon as you can see this 7 minute, it is a high phosphorylation of the MEP kinase. 15 minute, it is again, it is the similar. 30 minute, it goes down to the 24 hour, it diminishes. So, and then the problem start here. So when you do this further to quantify, you have to do it with the total protein. And then you need to strip this blot. And when you strip, and this is the problem, this is the part where you can, you are not sure result whether there are total protein exist here or it is due to this stripping, it get lost from the membrane. So there are various challenges, and we can discuss a little bit more on about the, just have have a look onto the cost, approximate cost of the each Western blot. So if you calculate, it is a rough calculation. Of course, this is, if you calculate the approximate rate, it's 500 crown, Swedish crown, it cost for each round. But we forget to calculate the number of hours we put into a Western blot. For example, it starts from the cell culture. It is the primary cell culture. You start, it takes 3 days to split, and then again you make more number of cells, and then you divide the cells, and then you put the VGF treatment, then harvest the cells, and all these things, it add up the time and time. And then you go to the Western blot, the membrane, and then you have this bubble problem, and then the stripping problem. So and you can add on the price on each hour. Approximately minimum price is 150 crown per hour for a research assistant. So number of hours, and then you can add the cost. So this is the conventional, but still most of the laboratory, like many laboratories across the world, they use this conventional Western blot. So what are the problems? The if you name it is the reproducibility. So when you do many times, like it does not reproduce because of there are factors. You cannot control the factors like the transfer rate or transfer to the membrane. Even if the same protein you do many times with this conventional Western blot, still you can have the problem of reproducibility. Then the quantification, it is another problem. This is the semi-quantitative. The time is another one. So, and one of the things is like if you have a more than 24 samples, we have generally 10, 12, or 15 samples we load. But if you have more than 24 samples or 30 samples, let's say, it is very difficult to run and control the like different among the different plots. And then another one is if you have a protein, target protein which have multiple phosphorylation sites, then it is very difficult to get this isoform. For example, you have the AKT protein where like AKT1, AKT2, AKT3, and all of them have like several phosphorylation sites. And if you try to map with this phosphorylation protein versus this total protein in this conventional Western blot, it is just kind of impossible to do this. And need more amount of samples. So at least you need the 10 microgram protein, total protein to charge it. And then stripping problem. So sometimes it is the 10 microgram is for many labs, it is okay. But if you see this, for example, the cancer biopsies or human patient samples, or you isolate the cells from the endothelial cells, for example, if you isolate from the mice, and you know how many mice, like you take the 6 month and then you have the little mice, and then you have to take out the lungs and then isolate the cells, and you have very limited samples, and that's where it is the amount of sample is so important to do the experiment. So you cannot afford to lose like if you do the conventional Western blot. So now the coming to the picture of this, whether the technology available for the other technology, yes, that nano all these sort of problems what I have discussed before in conventional Western blot. So let's see if did you know about this NanoPro technology before this webinar? Little bit of pause to take. If you know that, just write in the chat box yes, or if you don't know that, no, so that I I'll have a little bit more knowledge that if you know or if you don't know, let's take just 15-20 second break. Can I see the chat box here? Okay. Okay, I think it is the mixed response. I'm not sure that. But anyway, okay, coming back to the NanoPro technology, this is where I am going to little bit take 1 or 2 minutes extra to explain you, because some of you don't know the technology before. So this is the everything happens here in a capillary, very small thin capillary, and it takes only less than 0.5 microliter. I mean, half this capillary is like 400 nanoliter. So whatever sample it will take, so it can accommodate only 400 nanoliter. So if in the first step you take the lysate, it is from the either from the cells or the tissue, and then you do the protein, make the lysate, and then this lysate you have to mix with these ampolites and fluorescent isoelectric focusing standard, which is provided from the company. So ampolites, and this is the ampolite kind of it has to be, there has to be a kind of buffering system. So it is a similar thing like. You can say in the SDS page, it is a polyacrylamide gel. Uh, so in the first step, it takes this lysate, ampolites, fluorescent PI standard in mix, and then uh, it focused on the basis of isoelectric focusing. So wherever the PI of the protein matched, then it stopped the electrophoresis between this capillaries and then it will, all the protein moves according to their isoelectric point. Just to remember that this is not the depends on the charge size, so it is depending on the charge. So wherever this isoelectric point match, it will move from the positive end or negative end, cathode or electrodes anode side, but it will stop where this isoelectric point match with the pH of the pH of the protein. So depending on this, even if it is the similar size of the protein, it can move different direction or different side. After that, it is immobilized by the UV light, and then all the material which is be stick into the capillary, it is flushed out, and it will go to the take the primary antibody, then wash, then secondary antibody, and then wash again. Then here the ECL, Camille with ECL, and then you can get this light signal. And this light signal is captured by digital CCD camera, which is incorporated there. And you can get a peak kind of graph where you can see if the more protein is there, you can get the bigger graph, and it is the bigger peak. And if it is less protein is there, you can get the lesser peak. Okay, so I hope it is clear, but this is the representation of each capillary, and each experiment, it can accommodate like 96 capillary at a time. So it is huge number of, it is very high throughput technology. And then if you are still not clear, then this is probably this little video, then it can show how it happens in real terms. So here it is the small capillary, everything happens within this NanoPro machine, where it can take this, it's all automatic. So it takes the capillaries now. This capillary goes to the plate where you have the lysate and different samples you put here in this plate, and just take it at 12 capillary at a time. Now it is taking the sample, lysate and ampolites, and it loading the protein into this capillary. So after this, it goes to the electrophoresis separation plate, and it is going there. Here you can see this separation chamber, and now the electric current passed from the both sides, and you can see this separation of the protein from. So it depending on the isoelectric point of the each protein, it moves or it can call. Now it is the immobilization step, and then with the UV light, it gets fixed into the wall of the capillary. And after that, there is a step like primary antibody incubation, and then secondary antibody. This is the incubate with the primary antibody, and then it was again. So only difference is that we don't have the blocking step here as compared to the normal Western blot. Antibody, and then you can get the chemiluminescent signal here, and this is where you get the light, and then camera will capture. Okay, so sorry, yeah. So this is the kind of advantage of the NanoPro, where this I was describing this AKT protein, where you can see only this one or two band there, but if you do this NanoPro, you can get the several peaks. Even if you don't have the phosphorylated protein antibody, still you can figure it out, this phosphorylated part of the protein. And this is bit probably confusing for you because it is a complex experiment. But let's see, this is okay, this one. I think I just skip the slide. This is the conventional Western blot, where you can see these two bands. And but here is the part where you can see this NanoPro, and then you can get this double phospho ERK1, single phospho ERK1, double phospho ERK2. All the isoform what I was mentioning, it was not easy to detect in normal conventional Western blot. And then each peak is represented by this is the image which we can quantify exactly, like 27% or 18% or how much that situation. So it is easy to calculate. So the advantages are many. No gel run, no transfer, no stripping, automated, highly throughput, high reproducibility. One can accommodate 96 samples at a time. So if it is the pharmaceutical setting that you want to take or test the drugs, 96 different conditions, then it will be much more easier. So you don't have to do manually. Possibility of get the phosphorylated band without having a working phospho antibody. So many times we have the difficulties to get these phosphorylated antibodies. Still, it is possible with this instrument. Valuable samples, for example, biopsy isolated cells, that are crucial, and then you don't have to lose that samples. And these are the papers, some of the old papers where it actually started. This technology works, and it published in, and you can see, you can see the, it can get the 25 cells. You can get the signal from the 25 cells. So next, coming to the next, coming to the proximity ligation assay, and this is the one. This technology is developed in Uppsala by Professor Ulf Lendergren. He was the pioneer of the experiment, and he's still working. He has a lab here, and he developed this technology. So what is happening here is like when you detect, as you know, this the name itself, it says the proximity ligation assay. So when there are two proteins are close enough or in the proximity, and then it can detect the signal in much more like it is very sensitive technique. So if you see this first part, it is the two proteins are close enough, and then you have the primary antibody and secondary antibody, and here is a primary antibody, secondary. This primary and the secondary antibody is conjugated with a oligonucleotide, and then in the second step you add there are separate oligonucleotides, and if these two proteins are close enough, then these oligonucleotides will go and hybridize and to make a kind of circle here. If you see here, there is a circle, and this one part is hybridized to the one part of the oligonucleotide, which is it is called the plus strand or minus strand, hybridized to the other part of the antibody, which is conjugated to the other antibody. It's a minus strand. So these plus and minus strands make a kind of circle. So in this time, you have to add a ligase, and then this ligase will join this end, and it make a circle, and then it is a PCR reaction. After you put the tech polymerase and put all the nucleotides into that circle, and then this that's why it is also called, this is rolling circle amplification. If you see this other part of the other this part of the antibody, it amplifies so many times, and then it is very easy to detect the signal. And this smaller one, which is binding this is small oligos with HRP tag, so you can detect by the chemiluminescence, or it can be used for the fluorescent microscopy. It has a lot many applications, and this is the sensitivity is the main issue here. You can get so much high sensitivity even if it is in normal condition with the primary and secondary antibody. You cannot see, you can see it here in the RCA or PLA. So what we have done then to combine these two technology. In the NanoPro, you have the conventional like primary antibody, secondary antibody, and then chemiluminescent signal. So that is there. So more steps here. First step is ligation, and then second step is rolling circle amplification. You have here the normal PCR reaction going on, and then this HRP oligos hybridization in the last step. So these three steps has to be inside of this capillary or the machine. So as you know that this is the when you start with the thinking that this is our hypothesis, and we will do this, and then when you do real in practical things, that is not so easy, that you cannot do simply because of this. Okay, ligation, rolling circuit amplification, and oligonization. We have struggled so many years. It will, I think it takes us 3 years probably, because every time there is some problem. The machine was not able to start, cycle. So many things it has been there to troubleshoot before we went ahead and get some like interesting result. So let's see. This is the schematic diagram where this is the simple NanoPro, where you have the protein, then primary antibody, and then secondary antibody, where this this HRP is conjugated here, and then you get the signal. So here the NanoPro and RCA, and you can attach this secondary antibody plus strand and secondary antibody minus, and roll and roll and roll and roll. It can amplify so hugely. And then another situation is NanoPro plus PLA, which is PLA means this is the two side of each protein. So in this case, you have to take primary antibody from two different species. So that then secondary antibody, and then it is the one like the RCA. And directly this primary antibody attached to this to plus 10 or minus 10, and you can get, but always here there's two different antibodies. Okay, next, when we test this one, sorry, this combination, first thing we test it whether it does work or it does not work. So in the green line, you can see this phospho MAP kinase protein ERK1 and ERK2, PLA plus NanoPro, and here the blue line, here only simple NanoPro. And you can see that this is so much sensitivity. If you do this, NanoPro itself is a huge, the technology. It can give so much better signal than, because it is automated and then have several advantages. But if you add this PLA into this and look at the sensitivity, how much it goes up. It is so easy to it. And here you can see the quantification. Normal the PLA and NanoPro. So it was very good. Then we started. So then next question is this technology with this combination, whether this will work for this standard protein, the one protein, phospho arc or arc, this MAP kinase, or it also works for the other proteins. Okay, let's see this. And you can see this. We have tested a battery of protein here to check this, to validate this technology, whether it works for the other proteins or not. You can see here the first one, make one with different micrograms of protein. It was loaded, and then this first lane is NanoPro, and then second column is NanoPro plus RCA, and then you have this peak and then quantification here. And always you can see this quantification with normal. Whenever you add this PLA in combination with NanoPro, it has so much high sensitivity, several fold increase in terms of sensitivity. So it works for the another important protein, phosphosarc. Why it is the phosphoprotein is important? Because phosphoproteins are so much difficult to work with in any sense. Like it is so much in less quantity in a certain situation. That's why we were interested in to look in phosphoprotein, not the total protein or not the other protein. So if seems it works for the other protein as well. Then now phospho P38 and beta 2 microglobulin in the same way. So now the question is there any other application for this technology in combination? When you have the combination of technology, is there any other application? So it is a very interesting result. I can show you. This we take the human colorectal cancer patient sample from the benign. So you have no cancer 4 sample. So stage 4 means almost it is the metastasis stage where people are very in very advanced stages where they are almost like the chemotherapy and all sort of treatment, or it is very difficult for them patient to survive at this stage. But it is that in the cancer detection or cancer diagnosis, it is so important. If you diagnose it earlier, you can prevent it better. And main problem is one of the main problem is the diagnosis of the cancer in early. How do you diagnose? And that is one of the application here. So you can see this. If you have this green signal in the normal NanoPro, you don't see basically anything here in the benign state. In this stage 2 also, and then the stage 4 also, and it is so much increase with the sensitivity when you add this combination of this both technology. So you can check this phosphomyc one and then phosphosarc 418. And it is very interesting to see in this phosphorus arc. Here you can see this stage 2 and then stage 4. Here you have so much increase in phosphorus arc. But you cannot see in normal NanoPro. So you can only see this phosphorus arc increase activity in when you have the combination of this both technology. So it will be a great forward to see this diagnosis, and then of course you need to test many other things before it comes to the diagnosis part. But there is a potential application of this. So now it is the amplification part I have discussed now. But these two technology, does this have any role with this specificity of the protein detection? And that is one of the, because this is in the NanoPro and the PLA or RCA, in the RCA, whatever is there, even if the antibody, you know that many companies, they have this antibody and still it is so crappy, and then you don't know that whether this antibody is really giving you the right signal. But even if it in the RCA or NanoPro, even if a antibody it binds to the wrong target, then it can amplify, and you probably see and then see, okay, this is the this is the sensitivity, and you will be very happy. But that is not the case all the time. If it is the wrong and wrong places, wrongly it will bind this antibody, it can amplify so much, and then it will be confused or miss one. So if you do this two antibody at a time, two primary antibody, two secondary antibody, and do the same experiment, and then you can see that here it is we tested this normal NanoPro in this first one, and then this is the NanoPro plus RCA, then you can see the amplification very high. But if you do the two antibody, then only single peak. You can find only single peak. So now you discard all the peak. This is the only one which is we should focus on. And these are the other peaks which is not true. And you cannot see with this NanoPro with RCA if you use the one antibody, one primary antibody, and then so this is very important for to know the specificity of the protein. We check this aldose reductase, we check this S100A6, and you can see this one. And if you see this CDC34 protein, you can see this two proteins in the two peak here and the two peak in this NanoPro and RCA, and there is no peak. So these two peaks are not specific, even with the NanoPro and the NanoPro with RCA. So it can only be detected, the specificity, by this NanoPro and PLA. So in conclusion, the signal intensity is greatly increased by RCA as a standard NanoPro with the same antibody. And also the many isoforms could be detected. But the NanoPro and PLA can greatly improve the specificity of the protein detection because of this requires two different primary antibodies simultaneously recognize the same target. And that's why it is so much important. And now it is the kind of last slide here, and then I would like to acknowledge this. This is the Professor Lena Clason-Wells from Uppsala, renowned person in this field of vascular biology and angiogenesis cancer field, and where I was working this with her for so long time. And this is the Professor Uwe Lenderkin in Uppsala University, who was the pioneer, and both of them are still having the lab in the Uppsala University, and they are he's the one who developed this PLA or technology. This is Dr. Jun Hong from the Wolf Lenderkin's PhD student at that time, and we as the one who were collaborating closely and developed this combination of this technology, both NanoPro plus PLA, because she was the expert in PLA and I was the expert in NanoPro. So it was a great combination to mix these two people idea. Of course, it takes quite long time, but still, it is fruitful at the end. And Olink Bioscience is the one who is owning, I think, this PLA stops, and they provide a lot of sample to us to develop this technology, NanoPro stops. So I'd like to thank all of them. And thank you very much for, and if you have any concern or questions or anything, you can reach out to me with this email or LinkedIn. Yeah, now over to Dr. Susma. I think we have some questions probably.
Thank you, Dr. Narendra, for the wonderful presentation. We have a couple of questions in the chat which has come up. Dr. Rath has put up this question to you. What is the
Mhm.
sensitivity of this assay that you are talking about, and how much is the amount of protein required?
As I said in one of the slide, actually, it is the 25 cells it takes. Like it is depending on the what protein you are targeting and what antibody you have. But it is certainly it is from the picogram level you can pick this. This is, I think, it is from 2.5 something in the picogram level. At least you can see this sensitivity for phosphor protein. Is that clear?
Okay, there's another question from Dr. Rath, which he is curious about. How much cost effective is this technology? You know, when you compare it to Western blot technique, so how much cost effective is it?
That is a kind of, the subjective question actually, that the cost is like, if you do repeat many times with this, when I calculated the cost for the conventional Western blot, generally we forget to calculate the number of hours we put in each student or each researcher. And if you fail and repeat, and then actually at the end it gives similar cost actually. But only thing is, of course, cost is important, because this is the you have to buy the instrument probably, and then also the one of the major disadvantage, not all the antibody it works. So even if they have a certified antibody, still you need to test. All the antibody does not work, because most of the antibody they generated because of this, they recognize on in the conventional Western blot, because they are in unfolded form. And here most probably that antibody which is work for example the immunofluorescence or this immunohistochemistry, I think this is the in the more natural form. So that proteins or that antibody is more probably more work, it can work more in the nanopro setting.
There's another question from Anandita Paul, and her question is: can this technique be extended to diagnose genetic or metabolic diseases?
Yeah, of course, this is the good question. And then this is also that I think many pharmaceutical companies probably they are now, because that is the any protein based detection method can be used in this setting. So that is the beauty of this. And it is not the nanopro and PLA that has not has been used, but that is for definitely nanopro is using for the detection of the diagnostic company, detection of many diseases.
Okay. The next question that Dr. Parth Sarthi has put up is why it's NanoPro except PLA. Also, are these capillaries reusable, or we have to use new every time we use ampholytes?
No, it is the one of the benefit in this, when you combine this technology, because PLA reagents are also the expensive. It is not PLA reagents are expensive, and also the capillaries are, it is not good to use. They don't work well and then once you know that it is used, and it is, you cannot clean it perhaps, because it has only 400 nanoliter materials. And if it is the something, because you use the UV light and to the cross-link the protein there, so you are not sure that you cannot you can reuse this capillary. So it is recommended, I think, that you have to use this press, because that is the one, the capillary takes everything there. So if you have the uh, if, for example, you use the wrong capillary or bad capillary, then you are not sure, because this is so sensitive, and sensitive as well as that all the signal can be misrepresented or mislead.
Okay. So there's number of questions which are coming up, Dr. Narendra. So there's another question which Dr. Rath has asked again, whether the technology is additive. If we add more antibody, will it give you more accurate result? Is it like that?
Yes, you are asking basically whether it can be multiplexing or not, right? This is, you can add the many proteins, but before that adding many protein or many antibodies, then you have to be sure that which the protein is, you should be sure that the peak coming from the antibody is coming from the antibody. For example, here you can add like phospho antibody with the HSP70 or the beta 2 microglobulin, because it has only one peak but other proteins. So in that sense, you can definitely multiplex this technology. Yes.
Okay. Uh,
So each capillary can you can get more result from the each capillary with many antibodies. Yes.
um, there's another question is there any other technology that works in similar principle? And if so, how NanoPro PLA stands good against that technology?
This is, I'm not aware of this one, but I think there is another technology which came, and now it is it is just representing the just like Western blot. So it is the like kind of gel run, but it is in the capillary. So not, and there probably it is more that more antibodies probably it works. So not on the basis of isoelectric point, but on the basis of charge, on the basis of size. Sorry, on the basis of size, just like a normal Western blot.
Dr. Narendra, I have one question. Actually, I have many questions, but one of the very basic question is like, since this step does not involve the blocking step, okay, which we generally do it in Western blot. So, like, what about the non-specific binding or like non-specificity of interaction of the antigen and the antibody?
Yeah, very interesting question. That's how this is the. If you remember, or I can go back to the slide here. Uh, if you see this one, we know there are like four isoelectric point of this protein, or the four isoform of this protein. So you can see this one, two are phospho, double phospho ERK2, and then, but you don't see any other band, like, so other peak. You don't see any other peak. It is difficult to get the peak here. So still it is there. You can, of course, you can get the non-specific peaks, but here you need to validate. For example, if you take phosphatase here, and then do the minus phosphatase versus the plus phosphatase, and run the sample at the same time parallelly, and then you can see if the phosphatase will remove this signal. So that means you have phosphate group. So that's how you need to know the peak before you conclude something on the new protein, because this is a kind of new technology, and also you need to see the pattern, because it gives usually many peaks, and then it is, you have to map out which peak is coming from what. And also in one of the last slide, that I showed you, the specificity. One of the protein, I think it was NanoPro, it was giving 2 peaks, and then NanoPro plus RCA, it was giving 2 peaks. Again, if you see the join this NanoPro plus PLA, and then the peaks is gone. So that is specificity. Yeah, there are many different methods you can do it. Is that answer your question or something?
Yes, yes. Definitely. Another curiosity is like the peaks that you're showing here. Okay, so how do you know? Of course, you must have run standards and all. I think you run, and that's how you get to know. Okay, this peak is for the non-phosphorylated ERK. This is for the phosphorylated ERK. So how do you differentiate? How do you identify which peak is for which protein?
Oh my God, I should have worked for this company. You are so good question you are asking. And this is the question I was asking before to them. Like how you know that exactly specifically to this peak is coming from this protein. So what I have done or we have done during this standardization method, that we run one capillary assigned for the total protein, one capillary assigned for the phospho arc or arc 1 2 protein, and assigned for the single arc 1 protein or single arc 2 protein through antibody. Sorry, this all the antibody I am mentioning. And then you overlay all these peaks, you overlay, and then you know that this peaks is coming from arc 1, this peak is coming from arc 2, and this peak is coming from the total, and this peak is coming from the individual phospho arc 1 or 2. So that's how you map this which peak are specific to which part. And you also can confirm this by checking the isoelectric point. So always phosphoproteins are going to the left side or less isoelectric. So that's how you can figure it out. And once you map and once you standardize your peak pattern, then you can, I think, run. Then after some time you will know that this is the pattern for this protein. So it is much more easier after some time. But yes, in the beginning, it needs to be standardized a little bit.
Okay. So one last question from my side is since it's a new
Yes,
technology
yes.
and it's a very sensitive one, and it's like I feel it's not that cost effective as Western blot that we do in our labs here. So what is the you can say how many labs are like using this technology as on date? Like since it's a new one, so in India, I don't think like I'm not aware actually that any lab is using this NanoPro PLA technique for protein detection.
I think NanoPro and PLA, this combination has not come out. They are still working on, probably. I'm not sure how long it will take, because this is the uh, something IP rights or many things it come out. That has not been, because this is the research work we have done, and then it has, as a proof of concept, it works. So if the people, I don't know if, but this NanoPro, definitely many parts of the I think they are using in Germany, they are using in France, I know they are using in Finland. But because it takes time, that
Yes,
to
I know
get
it's.
the, like, scientists to get the confidence. So because Western blood, it is from 1970, I guess, and people are still using so much, because you can see directly their result, are minus or plus, or you can see the band or not the band. And because we have a mindset, Western blood, we know, everybody knows that Western blood, but it is so easy that, and instrument availability, and then price, and then reach out. So many things are there. But definitely this technology has much more powerful. But they have, I think, another one directly with the Western blood. Normal Western, but this one is directly with the NanoPro. I have not used it, but so basically, I like the all sort of technology, new one, because it saves so much time. And every lab, that, probably as a PI, you have to decide what instrument or what technology you have to do for this. But this is the one, when I see the papers like articles in the Nature and Science, and then all the normal Western blot they do, because that this is the quantification has so much in this one, then I think it is probably very accurate. At least I compare from the normal Western blot, do the quantification, and then I do it here in the NanoPro and do the quantification. It is much more, you can get the clarity. So it leads to much more get the clarity that you are saying the truth. Is there more question?
Yeah, I think there are no more questions, and neither from the participant side and nor from my side now, because I think that was a wonderful presentation, and I thank you once again, Dr. Pradhan, for agreeing to give us such a wonderful talk and enlighten us with this new technology which has come up for protein detection. And thank you once again to all the participants also for putting up such wonderful questions. And thank you all.
Thank you so much, Susma. It was a pleasure to talk to you, and it was so much interesting questions, so many. Thank you so much.
Thank you. Thank you, everybody.